Good morning, everyone. Okay, so today we are going to continue our lesson, okay, about protein as well as the structure and classification of protein. Okay, so proteins. Okay, now these proteins, they are complex macromolecules, macromolecules, okay, or polypeptides or polymers. Okay, so from the word complex macromolecules, it tells you that these proteins, they are big in size, and this explains why proteins, they cannot go through or bypass, okay, or diffuse. Okay, yeah, diffuse is the correct word. Diffuse. This tells why protein cannot diffuse out from our blood capillary. Okay, so they remain in our blood capillaries. Okay, because they are large in size. So proteins, they are complex macromolecules or polymers or polypeptides that are formed from amino acids. So meaning to say amino acids are the monomers of proteins. Okay, so protein, if you look at this diagram here, this general formula of an amino acid, right? If you look at this general formula of an amino acid, right? This tells you that an amino acid is an organic molecule because it has carbon, okay, as well as hydrogen and oxygen. So amino acid is an organic molecule that has asymmetric carbon atom, right? Asymmetric carbon atom called the alpha carbon. Okay, you can see the alpha carbon here at the center here. Okay, and this alpha carbon has an amino group, right? A carboxyl group, COOH, right? A hydrogen atom and a side chain. Okay, and this side chain, they are very different with each amino acid, and this gives the characteristic of that particular amino acid, right? Because of this side chain. Now this carboxyl group, right, and this amino group in an amino acid give the amino acid the amphoteric properties. Okay, amphoteric properties, meaning to say this amino acid can behave like acidic or can behave like basic. It can behave like acidic because the presence of the carboxyl group, and this amino acid can behave like base, right, or basic because of the amino group, right? And apparently this amino group and carboxyl group, they are always in ionized form, right? Ionized form. So this would be COOH, and then there will be a negative here, right? And then this NH2, there will be a positive charge here. Okay, so each amino acids, right, they are different because of the different side chain that I mentioned earlier. Okay, and because of this differentiation in the side chain, right, this gives the amino acids, right, different functional role in a polypeptide. Okay, and there are 20 in total of amino acids so far they are known here. Okay, 20 known amino acids. Right, now let's go on to classification of the amino acids based on their side chains. Okay, so apparently we have 4 different groups of amino acids. We have the hydrophobic, all right, hydrophobic. These are the hydrophobic amino acids. Then we have the hydrophilic amino acids, and then we have the acidic amino acids, and we have the basic amino acids. Now these hydrophobic amino acids, all right, their side chains, all right, are non-polar. You can see hydrogen, CH3, 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 CH3. And then we have this benzene ring, okay, benzene ring, right? And then CH3. So easy to identify, right? The non-polar side chains or hydrophobic amino acids. All right. So these hydrophobic amino acids, since they have non-polar side chains, so they're classed as non-polar amino acids, right? Non-polar amino acids that behave like hydrophobic, right? Now next, we have the hydrophilic amino acids. Hydrophilic amino acids, they have polar side chains, right? So they are grouped in the polar class, right? Now, how do you identify polar side chains? All right, very easy. Okay, like OH, OH, right? SHOH, right? And then oxygen here with the nitrogen here. Now, okay, the oxygen here, the oxygen atom here, in the polar, in the side chain of these polar amino acids, okay, they are not in ionized form. Okay, you can see OH, OH, okay, or other highly electronegative atom other than oxygen is sulfur, right? Sulfur, nitrogen, oxygen, these are highly electronegative atom. So, but this, although this oxygen here is highly electronegative, okay, this side chain here, they didn't exist, right? They didn't exist as negative charge here, right? It's OH, OH, okay? The same goes here, this sulfhydryl, right? The sulfur here, although sulfur is a highly electronegative atom, but it doesn't exist like a being charged here, okay? You don't see any negative charge here. You only see SH. All right, the same goes here, OH, NH2, NH2. All right, as well the oxygen here as well. It didn't exist as O negative or O2 negative here, or O negative, right? You only be seeing here NH2 and O. So examples of these amino acids in the polar class, they are serine, threonine, cysteine, okay, tyrosine, asparagine, and glutamine. And apparently only cysteine, all right, only cysteine, okay, cysteine is the only amino acid that has sulfur. Okay, cysteine, the only amino acid that has sulfur in the side chain or the functional group. When I say functional group, meaning to say at the end of the functional group. Okay, cysteine is the only amino acid that has sulfur. Okay, now examples of amino acids in the non-polar class, they are the glycine, alanine, valine, leucine, isoleucine, proline, tryptophan, okay, phenylalanine, and methionine. Okay, methionine has sulfur, but not as the functional group, right? In the long chain of the functional group, there is a sulfur there. Okay, different with cysteine. Cysteine has sulfur in the functional group. Okay, itu beza dia, cysteine dengan methionine. Alright, cysteine is the only amino acid that has sulfur in the functional group. Right, I rephrased that for you. Okay, next we already have the non-polar class, we have the polar class, and then now we have this acidic class, right? Acidic class. Right, now this acidic class of amino acid, they have hydrophilic properties, right? Very similar with the polar class. Okay, very similar. Both has hydrophilic properties. But the difference here is, right, for the acidic class, the side chain of the amino acid, all right, the the side group here, right, the functional group in the side chain of this acidic class, right, the oxygen atom there exists as ionized, right? You can see the O negative here and then O negative here for glutamic acid, and for aspartic acid as well, right? there is a presence of negative charge carboxyl group in the side chain. So meaning say the functional group of acidic class amino acid has negative charge carboxyl group, right? Compare with the ones in the polar class. They don't have the negative charge carboxyl group. Okay, now the fourth class is the basic class. Now for the basic class, okay, is very similar with the polar class where it has hydrophilic properties. Okay, but the difference here is, okay, there is a presence of positive charge amino group in the side chain. You can see here for histidine we have NH plus here. Okay, but we don't have that in the polar class amino acid. There's only NH2, right, which is for asparagine and glutamine. Okay, arginine, you can see the NH2 plus here. Okay, and lysine, you can see the NH3 plus here. Right, so there are 4 classes of amino acids, okay, based on their side chain properties. Now, properties of protein. Okay, many proteins, they are roughly spherical. You can see from here, right? Spherical. So this spherical, right? So these spherical proteins, they are known as globular proteins, right? While some others proteins, they are shaped, okay, like long fibers like this, and these are known as the fibrous protein. Okay, so for the STPM candidates, we have fibrous protein and globular proteins. These globular proteins, they are hydrophilic. Okay, they are hydrophilic and found in enzymes, some hormones, and hemoglobins. Okay, and these globular proteins, they are formed by compacted and folded amino acids. You can see this is a polypeptide, and these polypeptides, okay, are composed of amino acids as monomers. So they are being folded and compacted, right? They I refer to the polypeptides. Now these globular proteins, they are soluble in water. Okay, are soluble in water. That's why they are hydrophilic, because they are hydrophilic. Hydrophilic means it likes water. So globular protein soluble in water, but did not dissolve entirely, right? Although they dissolve, this globular protein dissolve in water, but it does not dissolve entirely. So since this globular protein does not dissolve entirely in water, this globular protein will form colloid. Okay, will form colloid when this globular protein is being dissolved in water or being mixed with water. You have a colloid. You don't have a suspension. You don't have a solution. No, you have a colloid. Okay, so colloid means insoluble particles that are suspended throughout another substance. So colloid, all right, this colloid will form salts with water. Okay, say if you add more water, you have salts, right? Now when the water in the salts are being reduced by heating, you will have gel. Okay, gel. And then this colloid, okay, remember colloid and colloid, when you have this globular protein dissolved in water, okay, when the water is being dried off, okay, you will have amorphous mass. Okay, amorphous mass. Amorphous mass means something that does not has a definite form. Tidak bentuk yang tetap. It follows the shape of the container. Then the colloid exhibits Tyndall effect. Okay, and the colloid also exhibits Brownian movement. So now fibrous protein, okay, they are hydrophobic. Okay, they are not hydrophilic, but they are hydrophobic, except fibrinogen. Fibrinogen, they are hydrophilic, right? And then these fibrous protein, they are found in skin, tendon, bones, and muscle. Right? This fibrous protein, as well as in your hair, keratin. Okay, your hair, it has keratin. Keratin is another example of fibrous protein. That's why your hair does not dissolve in water. Okay, because your hair is hydrophobic. Now, proteins are amphoteric in water, right, or zwitterions in water. Okay, and this is especially for amino acids with the acidic or basic side chains. Okay, remember the acidic and basic side chains, they have negative or positive charges in their functional group. Okay, so they can behave amphoteric in water. So now the carboxyl side chains, okay, meaning to say the acidic class of amino acids that has the carboxyl side chains in negative charges, right? So the carboxyl side chains will dissociate and behave like an acid in water. Okay. While the basic amino acid, all right, that has the amino side chains, the amino side chains will dissociate and behave like an alkali or base. So this is salt, right? When you have the globular protein dissolved in water, right? Yet some of the globular protein does not dissolve properly in that water, right? And then some of the water is being dried, you will have a gel. Okay. So this is the Tyndall effect, the scattering of light by colloidal particles. Okay, so you can see if you have a solution and then you hit the solution with a light, okay, the light will just pass through the solution. But for a colloid, you will be seeing a an area, okay, where the light is being scattered by the colloidal particles. Okay, now for globular protein, the light is being scattered by the globular protein here. Okay, so this is the amorphous, all right, mass, right? So meaning to say globular protein, okay, when the water in the colloid is being dried out, okay, you will get an amorphous mass. Okay. Now, protein also act as a buffer to resist pH changes when a small amount of acid or alkali is being added. Okay. At isoelectric point, okay, isoelectric point means to say the total of charges in equilibrium at certain pH. Means to say at certain pH, it can be 7, it can be 8, or it can be 5. All right. The charges, okay, of a molecule, all right, will be in equilibrium with the other charges of other molecule in that environment. Right, so at isoelectric point, proteins, okay, are least soluble in water, okay, because of the intermolecular bonding of the COO negative carboxyl group, okay, and the amino group NH2+. So if pH solution is above its isoelectric point, right, above its isoelectric point, so meaning to say more charges in the environment, so the proteins will be negatively charged and move towards a knot. If the pH solution is below the isoelectric point, proteins will be positively charged and move towards cathode. Okay, now polymerisation of amino acids, okay, means say the formation of polypeptides. So the amino and carboxyl groups, okay, you can see the amino and carboxyl groups on a pair of amino acids undergo dehydration reaction to form a covalent bond called peptide bond. So you can see we have two amino acids here which acts as monomers, right, of a polymer. Now for this example, in this diagram, okay, we have two amino acids. Okay, since we have two amino acids, we will get a dipeptide. Okay, so in the polymerization of dipeptides, okay, polymerization of a dipeptide, the amino, okay, the amino and carboxyl groups, right, on a pair of amino acids undergo dehydration reaction. So dehydration means say a water must be removed from the monomer, okay, from the substrate. These are the two substrates here. Air mesti dibuang. Okay, and when this water is removed, a covalent bond will be formed between these two amino acids. Now let's take a look here on this amino acid here. The OH here from the carboxyl group will be removed, and this OH will combine with the hydrogen from the amino group. Okay, so we have H2O. Now between this carbon and this nitrogen, right, this carbon and this nitrogen, a peptide bond will form. So you can see here, right? So this particular carbon, the OH is already removed, and for this particular nitrogen, the hydrogen is already removed. So we have a peptide bond here. So the two amino acids linked by peptide bond are not free to rotate around the nitrogen carbon linkage. Okay, so meaning to say yang carbon dengan nitrogen ni dia tak boleh pusing-pusing. It fixed on a plane. Okay, plane, dia tetap. Now because this peptide bond has a partial double bond character, right, this lack of rotation determines the structural character of the coils and other regular shapes formed by chains of amino acid. Now means because of this peptide bond that does not allow free rotation of this carbon and nitrogen, right, this peptide bond will determine, right, the structural character of the type of proteins. Okay, you will learn on that later on. All right, because we have beta plated sheets, we have alpha helix. These are the structural some of the examples of the structural types of the protein, right, is being caused by this peptide bond. So a protein is composed of one or more long unbranched chains. Okay, so each chain is called a polypeptide and is composed of amino acids linked by peptide bonds. So a dipeptide, okay, or any polypeptide, right, will still, okay, has an amino end, this is the amino end, and a carboxyl end. Okay, so just want to show you, okay, the OH from the carboxyl combined with the hydrogen from the amino group of another amino acids. Okay, and then the carbon from the carboxyl group will form a peptide bond with the nitrogen from the amino group from the other amino acids. Okay, now note, okay, please take note, right, in this example of polypeptides, okay, there are three amino acids here, okay, with two peptide bond. You know, please take note that the carbon, all right, from the C terminus will form the peptide bond with the N terminus from another amino acid. Okay, so apparently meaning to say, all right, one end of an amino acid that has the carboxyl group is known as the C terminus. Okay, C refers to the carbon here for the carboxyl group, and the other end, right, the other end of an amino acid, right, that has the N here, the nitrogen here, is known as the N minus. Okay, or if we go back here, right, apparently the C minus will form a peptide bond with the N minus of another amino acids. Now, structure of proteins. Okay, so the shape of a protein determines its function, right? Shape of a protein determines its functions. So the structure of proteins consists of 4 levels. We have primary structure, secondary structure, tertiary structure, and quaternary structure. Right, primary structure. Okay, this is the basic structure of a protein, right? So the primary structure of a protein is its amino acid sequence. So the R groups that distinguish the amino acids play no role in the peptide backbone of the protein. So a protein can consist of any sequence of amino acids. So a protein containing 100 amino acids could form any of 20,100 different amino acid sequence. So let me rephrase that, right? A primary structure of polypeptide, all right, it has only sequence of amino acids, and these amino acids, they are not being folded, overlap or anything, right? They're just being arranged side by side. No interactions between their side chains. So you have a linear group of amino acids. That is the primary structure. Okay, so to easily define a primary structure of protein, right? So a primary structure of protein is the amino acid sequence in a protein. So now an example of a primary structure of protein, right, is the hemoglobin, that composed of 2 alpha globin peptide chains and 2 beta globin peptide chains. So the alpha globin chains differ from the beta globin in the sequence of the amino acids, right? You can see from this diagram here also, this sequence of amino acids, they are very different based on their side chains. Any alteration in the normal sequence, meaning to say if this part of amino acid is being removed and replaced with another amino acid who shouldn't be here, right, this will have drastic effects on how the protein will function. Okay, so meaning to say the sequence of this amino acid in the primary structure of a protein is very important to be in orderly manner, right? Now please note that this sequence of amino acids, right, they do not interact to form a primary structure. But if this sequence of amino acids, they interact with each other, okay, they will form another different structure. Okay, now the second structure is known as secondary structure, okay, that shows hydrogen bonding patterns. So the amino acid, right, the amino acid side groups and peptide groups of the main chain can form hydrogen bonds with water or with other peptide groups. Right now, meaning to say, you know that amino acids has hydrogen, right, on their side groups. They have many hydrogen. So this hydrogen can form bonds with water or with other peptide groups. So secondary structure, okay, of this protein is resulted from the hydrogen bonds that form between nearby amino acids. Hydrogen bonding between amino acids causes the polypeptide to form an alpha helix or beta pleated sheet. So now in this diagram here, you can see here, this hydrogen here, right? This hydrogen here bond a hydrogen bond with the oxygen of another amino acid in another polypeptide, right? So because of that, this polypeptide, it is being folded. Dia berlipat-lipat, nampak kan? Okay, then this oxygen here bond hydrogen bond with this hydrogen from another amino acids, and the story goes on. So because the formation of the hydrogen bond between nearby amino acids, we have beta pleated and alpha helix. In the alpha helix, you can see as well the oxygen here bonds form the hydrogen bond with this hydrogen of another amino acids. Okay, the same goes here. So hydrogen bonding between every fourth amino acid, all right, forms the spiral shape of the alpha helix. So we get a alpha helix, or a secondary structure of a polypeptide if the amino acid form hydrogen bonds with another amino acids on every fourth amino acid. Okay, or let me rephrase that. We get a secondary structure of polypeptide if there is a formation of hydrogen bond between every fourth hydrogen between every fourth amino acid, sorry, right? Or let me rephrase it again. Oh my goodness, we will get a alpha helix structure of polypeptides if there is a formation of hydrogen bond on every fourth amino acid, right? And then for the beta plated sheet, right, another form of secondary structure can occur between regions of polypeptide aligned next to each other. So if the polypeptide are being aligned next to each other, hydrogen bond will form between the amino acids. Okay, and this structure of polypeptide is known as the beta plated sheet. So the aligned polypeptide can be either parallel or anti-parallel. So these two types of protein secondary structure will create regions of the protein that are alpha helices, okay, and beta sheets. Alpha helices means to say the regions of the protein are being cylindrical, right? And for the beta pleated, okay, the regions of the protein will be planar. So this is a planar, okay, and this is cylindrical. Okay, now a protein spinal structure can include regions of each type of secondary structure. Okay, so now example here, the DNA binding protein usually have regions of alpha helix that can lay across DNA and interact directly with the bases of DNA. And then porine proteins that form holes in membranes are composed of beta sheets arranged to form a pore in the membrane. Okay, these porine proteins you will come across in the third semester. Okay, this DNA binding protein you will come across in the third semester as well. Okay, and then in hemoglobin, the alpha and beta globin peptide chains that make up the final molecule each have characteristic regions of secondary structure. Okay, I will further explain this later. Now, the fibrous protein, right, exists as helices or plated sheets. Examples are keratin, which is a protein in your hair, and silk. Okay, both of these silk and keratin have only a secondary structure. Okay, so if you're wondering which types of protein, right, that has secondary structure polypeptide, it is the keratin as well as the silk. Okay, now we have a tertiary structure of protein. Right, this is when the polypeptides are being folded and links to each other. Now, the final folded shape, right, or 3D shape of a globular protein is called the tertiary structure. So, this globular protein, they tend to ball up into rounded shapes. So, this tertiary structure contains regions that have secondary structure. So now, a tertiary structure protein has two or more secondary structure protein. You can see this is the beta pleated sheet protein. They are in planar arrangement, right? This is the alpha helix polypeptide because they are in cylindrical. This is the alpha helix. This is the beta pleated sheet. This is the alpha, beta, alpha, alpha, beta, and beta. Okay, so now to sum up, okay, there are four types of protein structure. Okay, so we have the primary structure where the amino acids are being arranged in sequence, and then we have the secondary structure, right, where we have the beta pleated and alpha helix, and then we have the tertiary structure, okay, where you have combination of this secondary structure of proteins. So now a protein is driven into its tertiary structure, right, by these three factors. Hydrophobic exclusion from water. Okay, you can see in this diagram here, this polypeptide, they tend to ball up, right, because they want to stay away from the water, right, and we call that behavior as hydrophobic exclusion. So because the environment here is watery, and apparently the inner part of this polypeptide, right, they don't have water here, so this region is free from water, and since there are interactions between the non-polar groups, so we have hydrophobic exclusion. Then we have ionic bonds between oppositely charged R groups that bring regions into close proximity. Okay, so this is the ionic bond, meaning to say this particular amino acid, okay, that has the acidic or basic functional group will form an ionic bond with another amino acid that has, okay, that has the negative or positive charge in the functional group, right? So positive and negative, we have an ionic bond here. Okay, and another is disulfide bond, right, or disulfide bridge, okay, that lock particular regions together. So I mean to say, in this one polypeptide, okay, apparently the cysteine, right, of this region here and another cysteine of this region here, okay, will form a disulfide bridge between the sulfur in the functional group from both cysteines. So this tertiary structure, right, is stabilized by a number of forces. Okay, so the four forces, they are hydrogen bonding, right, that is between the R groups of different amino acids. Okay, we have a diagram here for that. And then the electrostatic attraction, okay, between R groups with opposite charge. You can picture that, right? It's more or less like this. Okay, they don't have the ionic bond, but then they have the electrostatic attraction. Then hydrophobic exclusion, right, like here from the non-polar R groups, and as well as covalent bonds in the form of disulfides. Okay. Okay, now class, the electrostatic attraction between the R groups with opposite charge. Okay, let me define this, rephrase this. Okay, this refer to the van der Waals attraction. Okay. Okay. Sorry class, electrostatic attraction between R groups with opposite charge, right, is similar with ionic bond. Okay, but not ionic bond. Okay, so there is another factor here, E. E refer to the van der Waals attraction, right? You need to say attraction or forces of attraction between two non-polar functional group of two amino acids. Now, the stability of a protein, right, once it has folded into its tertiary shape, is influenced by the attraction within the interior of the protein. Now, examples. Okay, two non-polar chains in the interior that are very close together are attracted by the weak van der Waals forces. So these weak van der Waals forces can form strong attraction when are many, because no holes or cavities exist in the interior of the protein. Can you picture that? Okay, this van der Waals attraction is very weak, but when many amino acids, right, form this van der Waals, okay, with another amino acids, okay, when these van der Waals are many, so it becomes a strong attraction, right? So the variety of different non-polar amino acids with different size of R groups with its own distinctive shape allows non-polar chains to fit precisely within the protein interior. Changing a single amino acid can drastically alter the structure and the function of a protein. Okay, now example, right, is the sickle cell hemoglobin, right? It's a change of a single glutamic acid for a valine in the beta globin. So this change substitutes a charged amino acid for a non-polar, okay, on the surface of the protein, leading to the protein to become sticky and form clumps. Another variant of hemoglobin, right, called HBE, is a change from the glutamic acid to lysine at different site in the beta globin chain. So the structural change resulting in blood disorders called anemia and thalassemia. Meaning to say, do you recall the if you recall the primary structure of the amino acids, where we have the amino acids are being arranged in sequence. If one of that particular amino acid is being replaced, for example, right, we have a glutamic acid. The glutamic acid in that sequence is being changed to lysine. So what happens, right? The beta globin chain, right, will change its shape. It won't behave in the beta globin chain, right? So this in turn will change the whole shape of the hemoglobin. So the structural change will result in blood disorder called anemia. That's why in anemia, right, this shape of the red blood cell is not spherical, but instead like a sickle. Okay, now quaternary structure, all right, the fourth structure of a protein, okay, or polypeptide. So quaternary structure is the overall protein structure that results from the aggregation of the polypeptide subunits. Okay, quaternary structure is only found in proteins with multiple polypeptides. The final structure of the protein is the arrangement of the multiple polypeptides in space. So in a quaternary structure of a protein, right, meaning to say that particular protein has many polypeptides, right, many polypeptides, right. When I say many, you can is more than 2 or 3. So now example is collagen. All right, collagen is fibrous protein that has three, you can see here there are three identical helical polypeptides that intertwine into a larger triple helix, okay, giving the long fibers great strength. So this suits collagen fibers to their function, okay, as the girders of the connective tissue in skin, bone, tendons, ligaments, and other body parts. All right. So this is how the collagen look like in cellular level. Okay, and then we have hemoglobin. Okay, so a hemoglobin consists of 4 polypeptide subunits. Okay, 4 polypeptide subunits, 2 alpha polypeptides and 2 beta polypeptides. So both alpha and beta subunits consist primarily of alpha helical secondary structure. Okay, so each subunit has a non-polypeptide component called heme. Okay, you see the heme here that has an iron atom that binds with oxygen. All right, that's how we get the hemoglobin. When this we have the oxyhemoglobin. So one hemoglobin has 4 hemes with 4 iron, right? And each of these iron can bind with an oxygen. So one hemoglobin will form an oxyhemoglobin, which consists of a hemoglobin with 8 oxygen atom, right? Because one iron can bind with an oxygen, O2. So 2, 4, 6, 8. So for oxyhemoglobin we have HBO8. So recap, this is the primary structure, which is the sequence of the amino acids, right? And when these amino acids interact with each other, we have the secondary structure, which is the alpha helix in cylindrical or the beta pleated sheets, right? And then from the secondary structure we have the tertiary structure, okay, and from the tertiary structure we have the quaternary structure. So a quaternary structure, it has many either tertiary structure or many secondary structure, okay, in one quaternary structure. Okay, this to show you the interaction between the amino acids in a quaternary, tertiary or secondary structure. Okay, now chaperonin, all right, or chaperone proteins. These proteins, all right, that cannot function properly unless they are being folded into their correct or normal shape. Okay, cell contains chaperone proteins which help new proteins fold into their normal shape or correct any misfolding of a new protein. So meaning to say, chaperonin, right, is sort of like a structure that helps the protein, all right, to fold properly. Okay. So now example, cystic fibrosis. Okay, they are associated with misshapen of the protein because of the missing or malfunctioning of chaperone protein. Okay, so you can see here, this is a chaperonin, right? The polypeptide enters the chaperonin, right, and then it's being assembled in its shape, and then the correctly folded protein will leave the chaperonin. Now, classification of proteins based on structure, composition, and function. So when it comes to structure, we have globular, fibrous, and intermediate. When it comes to composition, we have simple and conjugated. Okay. So if based on structure, again we have the fibrous, globular, and intermediates. So for intermediate protein, right, they are the fiber-shaped polypeptide chains. Okay, they are fiber-shaped and they are soluble in water. By right, fiber-shaped or fibrous protein, they are not soluble in water. Only the globular protein are soluble in water. But for intermediate protein, they are soluble in water. Okay. An example of this intermediate protein are fibrinogen, okay, which is important in the blood clotting. Now, if based on composition, we have the simple and conjugated. Right, for simple proteins, okay, they are proteins that contain only amino acids. Okay, example like albumin, globulins, okay, histones, and scleroproteins. For conjugated, okay, for conjugated, they are protein, right, that has prosthetic group. Okay, so we have phosphoprotein, okay, which means to say protein that has phosphates. We have glycoproteins, protein that has carbohydrates. We have nucleoprotein, protein that has DNA. Okay, example is like the chromatin. Then we have chromoprotein, we have lipoprotein. Okay, lipoprotein when protein is being added with lipids. Okay, then we have flavoprotein, proteins that are being added with FAD. FAD we learn later. Okay, in the electron transport chain. Okay, then we have metalloprotein when protein is being added with metal. Okay, so most globular protein, they are enzymes, right, as well as antibodies and hormones. Okay, fibers, they are collagen and keratin. Okay, and conjugated, they are hemoglobin. Now, in this conjugated protein, for metalloproteins, one of the example is the hemoglobin, right? Because hemoglobin, is a protein, right, that has the iron in the heme, right? So hemoglobin is one example of the conjugated protein. The next classification of protein based on functions, right? So we have enzymatic proteins, right? These are the proteins that selectively accelerate chemical reactions. Okay, examples are digestive enzymes that catalyzes the hydrolysis of bonds in food molecules. Okay, so example enzymatic proteins like protease, lipase. Okay, all those enzymes are SS behind. Then we have defensive proteins. These are proteins that give protection against disease. Examples are antibodies that inactivate and help destroy viruses and bacteria. So antibodies, these are defensive proteins. You will learn this in detail in semester 2 later. Then we have storage proteins. Okay, means say proteins that can store amino acids. Example is casein. Okay, casein. Then we have transport proteins. This you will learn in detail as we progress in semester 1. Okay, transport proteins. So these transport proteins transport substances like hemoglobin, for example. Hemoglobin transport oxygen. Okay. Then we have GLUT4 transporter. Okay, GLUT4 transporter is a protein pore, right, that can be found in the membrane of the hepatocytes, our liver cells. This GLUT4 transporter transport glucose, right, from our blood into the liver cell. Then we have hormonal proteins. Okay, proteins that involve in the coordination of an organism activities. For example, is the insulin. Insulin is a protein, right? It's a hormonal protein, right? That will cause, right, the uptake of glucose from the blood into the liver cell, right? And then we have receptor protein, protein that responds to cell, okay, because of the chemical stimuli. Or let me rephrase that. Receptor proteins, okay, are proteins that response of cell, okay, that give response of cell to chemical stimuli. Okay, so one example of receptor protein, okay, is this acetylcholine receptor. Acetylcholine receptor, right, which bind with the acetylcholine. Okay, this you will learn in more detail in semester 2. Okay, and then we have contractile and motor proteins, okay, which is the actin and myosin. that function in the movement, okay, or contraction and relaxation of a muscle fiber. Okay, actin and myosin. Actin and myosin also you will learn in detail in semester 2. Okay, so for your level now in semester 1, just know that actin and myosin are example of contractile and motor proteins. Right? And then we have structural proteins. For example, is the keratin that gives the hair, alright, its structure. Gives the hair's framework. Okay, structural framework. Yeah, that's more better. Okay, now the last part for today's lesson, right, is the denaturation and renaturation of protein. Okay, so denaturation is a process, okay, you can see from here, which a protein loses its native shape because of the disruption of the weak chemical bonds and interactions, thereby becoming biologically inactive. Proteins can be denatured, okay, denatured. Oh my, proteins can be denatured when the pH, temperature, or ionic concentration of the surrounding solution changes. Okay, transferred from an aqueous environment to a non-polar solvent, and when exposed to chemicals. So these are the conditions that can cause proteins to be denatured. Right? Okay, now when it comes to pH, right, excessive hydrogen ions or hydroxide ions will break the ionic bond and hydrogen bonds in the protein structure. We recall the secondary and tertiary structure of the protein. Okay, then in temperature, excessive heat will agitate the polypeptide chain, all right, enough to overpower the weak interactions that stabilize the protein structure. Okay, and in ionic concentration, what happens? The excessive ions will break the ionic and hydrogen bonds in the protein structure. Okay, and in non-polar solvent, what happens? The polypeptide chain will refold. Okay, so that the hydrophobic region will face outward toward the non-polar solvent. By right, the hydrophobic will stay in the center of the protein. Okay, but in non-polar solvent, what happens? The polypeptide chain will refold, right? So that the hydrophobic region face the non-polar solvent. So hydrophobic interactions of the polypeptide are disrupted. Now in chemical structure, what happens? The chemical will disrupt the hydrogen bonds, ionic bonds, and disulfide bridges that maintain a protein's shape. Okay, I don't have the diagram here, but you have the diagram in the previous slides, right? Now, denatured proteins are usually biologically inactive. Okay, so denatured enzymes, example, denatured enzymes cannot catalyze any enzymatic reaction, right? And then the traditional methods of food preservation, salt curing, and pickling involve denaturation of proteins. So foods are kept in a solution containing a high concentration of salt or vinegar, which will denature the enzymes of most microorganisms and prevented the microorganisms from growing on the food. Okay, now renaturation is the reconstruction of a protein to their native shape after denaturation. Okay, so after a protein is being denatured, we can renature the protein. Example, a heat denatured DNA can revert to its native shape by cooling slowly the two strands, right, which will reform into its native double helix strands. Now, not all proteins can be renatured, right? Not all proteins. Okay, so that's it for today, right? Okay, see you in the next lesson. Bye.